Hello students, welcome you all to a new video of Sunanda's tutorial. In this video, I will discuss about the plant growth hormones, photoperiodism and phototropism. It is present in your SSB, TGT, OPSC, PGT and SSB lectureship syllabus. So let's start. What is plant growth regulators? These are small organic compounds of diverse chemical composition which affects or influence the physiological activities of the plant at a very low concentration. Plant growth regulators are also called plant hormones or phytohormones or plant growth substances, PGS. On the basis of their function in a living plant body, PGRs or phytohormones are classified into following groups. What are those groups? One is plant growth promoter in a positive sense and one is plant growth inhibitor. Plant growth promoters are involved in growth promoting activities. They promote or they have a positive effect on the plant, like cell division, cell enlargement, tropic growth movements, flowering, fruiting, seed formation, etc. And the example of these plant growth promoters are auxins, gibberellins, cytokinins. Then plant growth inhibitor, they affect adversely to the plant. They involved in growth inhibiting activities like dormancy and abscission. For example, abscisic acid and ethylene. These are the examples of plant growth inhibitors. Then the first one is auxin, which is a plant growth promoter. At first, the discovery, auxin was first phytohormone to be discovered. Charles Darwin and his son Francis Darwin observed that the tip of the coleoptile of canary grass responded to unilateral light by bending towards the light source. So, he took the coleoptile of canary grass of a plant and its shoot tip. Then he provided a unilateral light on it. After some days, he observed the bending of this coleoptile towards the light source. And this experiment was first performed by Charles Darwin and his son Francis Darwin. Later on, it was found that the tip of the coleoptile is a source of auxin that caused the bending of the coleoptile towards the light source. Then F.W. Wendt in 1928 isolated auxin from the tip of the coleoptile of oat, Avena sativa seedling. The most commonly occurring naturally occurring auxin is indole acetic acid that resembles to amino acid tryptophan from which it is synthesized. So, indole acetic acid synthesized from the tryptophan. Then indole acetic acid synthesized and transported basipetally, that is from apex to the base of the plant through polar transport. Polar transport means it transports from one pole to other pole of the plant and by the parenchymatous cell or tissue. And they also travel by non-polar transport by the phloem. The synthetic auxin or man-made auxins are naphthalene acetic acid, indole butyric acid or 2,4-dichlorophenoxyacetic acid 2,4-D and 2,4,5-trichlorophenoxyacetic acid 2,4,5-T. What are the physiological effects of auxin? First one is auxin stimulates cell elongation. It controls cell division in vascular cambium and stimulate xylem differentiation. The differentiation of xylem into the other components of xylem. It prevents the premature abscission of leaves, flower and fruits. In higher plants, apical buds where the auxin actually produce suppress the growth of lateral buds. That means the auxin actually suppress the growth of the lateral bud. And this phenomenon is known as apical dominance. Removal of apical bud or decapitation causes the sprouting or growth of the lateral buds. The decapitation method is widely applied in tea plantation and haze making. Then auxin promote the growth of the stem and coleoptile but inhibit the growth of the root. It only initiates the root growth but cannot help in the growth of the root. It only initiates the root formation. Then it causes the bending of coleoptile towards light, and that phenomenon is known as phototropism. The bending or movement of the plant upper part towards the light source is known as phototropism or tropic movement of the plant. And controlled geotropism. What is geotropism? The growth in response to gravity. Root generally shows positive geotropism. Then uses of auxin. Auxin are used to initiate rooting in stem cutting, exogenous application of naphthalene acetic acid and 2,4-dichlorophenotic acetic acid induces flowering in pineapple and then 2,4-D, which is dichlorophenoxyacetic acid, and 2,4,5-T, which is 2,4,5-trichlorophenoxyacetic acid, togetherly used as herbicide to kill dicot weed, but it does not affect the monocot plant. Both IAA, that is indole butyric acid, indole butyric acid, and the IAA, which is the indole acetic acid, togetherly used to produce seedless or parthenocarpic fruit in tomatoes. Banana is a natural parthenocarpic fruit because it contains high auxin. Then the gibberellin. Gibberellin was first isolated from the fungus Gibberella fusiculae, which is a fungus that causes bacchane or foolish seedling disease in rice. At present, more than 110 naturally occurring gibberellic acid are known, though many does not have any biological activity, and they are referred or designated as GA1, GA2 or GAN like this. The most common gibberellin is GA3, which is gibberellic acid 3. Chemically, GA are tetracyclide diterpenoids consist of 4 isoprene units, and the chemical formula of it is C5H8. It is acidic in nature, so known as gibberellic acid. Then gibberellic acid synthesis occur in young leaves, buds, developing seeds, fruits and in roots. It transported passively through xylem and phloem. They transport by non-polar method. Physiological effects of gibberellic acid: it causes extension of stem due to cell elongation, not by cell division. It only helps in cell elongation. In germinating seed, embryos secrete gibberellic acid that promote the production of amylase and other hydrolases necessary for mobilization of reserve food in germinating seed. So it helps in seed germination. Then it causes the increase in the length of the internode, and the process is known as bolting in rosette plant. And exogenous application break the dormancy of seed as well as in bud. Exogenous application promote flowering in long day plant. Later on I will discuss what is long day plant and short day plant. And it induces parthenocarpy, that is from production of seedless fruit, improves shape of the apple. Then it promotes the formation of pistillate, that is the female flower in maize, but promotes staminate, that is male flower in spinach, cucumber and hemp. Then it is a substitute of vernalization. Vernalization means low temperature treatment for flowering. Then commercial GA3 is used to increase stalk length of seedless grapes and allow grapes to grow larger. Then It also used in malting of barley in brewing process. It delays senescence of citrus, increase the height of sugarcane and sugar yield. Then fern prothallus, which is the fastly formed gametophyte of fern, releases GA3 derivative that promote the development of antheridia on adjacent gametophyte. Here it acts as a pheromone. Then cytokinin. At first, Sook and his co-workers found that the tobacco callus could proliferate only if the nutrient medium contains extracts of yeast or coconut milk or DNA in addition to auxin. Then Skoog and Miller identified and crystallized the cytokinesis promoting substance called kinetin and or N6-phosphorylamidopurin from the autoclaved herring sperm DNA. Kinetin does not naturally occur in plant. Chemically, cytokinin are purine derivative similar to adenine. Zeatin, which is the first isolated cytokinin from kernel of maize grain and liquid endosperm of coconut. In all plants, zeatin or benzyl aminopurine BPA is most common naturally occurring cytokinin, but dihydrozeatin and or isopentylene adenine are the most commonly found in higher plants as well as in bacteria. The physiological effects: it stimulates cell division or cytokinesis, causes enlargement of stem cell rather than longitudinal extension. It helps to overcome apical dominance and promote the lateral bud growth, promote formation of new leaves and chloroplast. It promotes the nutrient mobilization from other parts of the plant to leaves, which helps in delay of the leaf senescence, and this is known as Richmond-Lang effect. Cytokinin increases number of buds in funaria. It also helps in conversion of Morse protonema to leafy gametophore. It releases the dormancy of seeds and buds, induce flowering in short day plant, increase the resistance of plant to adverse conditions such as low and high temperature and to certain diseases. High concentration of cytokinin induces epinasty, that is downward curvature of leaves. Then ethylene. HH cousin confirmed the ripened orange releases a volatile substance. Ethylene is the only gaseous hormone, and at first cousin confirmed its presence in the ripened orange, and he also concluded that this substance or this volatile gas hastened the ripening process of banana. Later on, this volatile substance was identified as ethylene or ethene, CH2 double bond CH2, a gaseous phytohormone. Then again in 1934 established that ethylene is a natural plant hormone responsible for fruit ripening. The precursor of ethylene biosynthesis is amino acid, which is methionine. It is very important point. Then the physiological effect of ethylene. It promotes fruit ripening and synthesis, accelerate the abscission of plant organ like flower, fruit. In the abscission zone, ethylene promotes weakening of cell wall. Then it enhances the respiration rate during the fruit ripening. This rise in rate of respiration is known as respiratory climacteric. Then breaks the seed dormancy of cereal and bud dormancy of potato tuber. It initiates germination in peanut seeds and sprouting of potato tubers. It induces flowering in mango, pineapple, but inhibit flowering in most of the cases. So then it promotes the root growth and root hair formation by help of the plant to increase the absorption surface. Then it promotes the female flower in cucumber. Ethylene in high concentration induces the adventitious rooting and root hair formation. It induces leaf epinasty, that means downward movement of the leaves, curvature, leaf curvature. Then ethiphone, when spread absorbed by the plant and releases ethylene slowly. This is commercially used to enhance fruit ripening, abscission of flowers and fruit. Then the last one, which is abscisic acid or stress hormone. 3 independent researchers reported 3 kinds of growth inhibitors like inhibitor B, abscissin 2 and dormin. Latter all were found to be chemically similar and named as abscisic acid. Abscisic acid is the natural growth inhibitor synthesized in all cells of vascular plant and mosses that contains plastid. Abscisic acid is transported through xylem and phloem. Chemically, this abscisic acid is 15 carbon terpenoid compound which is known as sesquiterpenes that derived from the terminal part of the carotenoid precursor. And they have some physiological effect like they promote dormancy. Then the dormancy of seed is broken only when ABA level is overcome by gibberellins. ABA inhibit the gibberellin mediated alpha amylase formation in germinating cereal grains and thus inhibit their sprouting. So it have the anti-gibberellin activity. It promotes abscission, that means premature leaf and fruit fall. It causes the leaf senescence by stimulating destruction of chlorophyll, inhibiting protein and RNA synthesis. It induces stomatal closure by inhibiting K plus ion uptake into guard cell and by stimulating malate leakage. Stomatal closure occurs during water stress or drought condition, water logging condition and other adverse environmental condition. ABA increase the tolerance of plant to various kind of stress. Hence it is called as stress hormone. It acts as antagonist to gibberellic acid hormone, so known as anti-gibberellin. So it is all about your different types of plant growth hormones and their physiological activities. Now we will study about the photoperiodism. What is photoperiodism? The length of the light period is called photoperiod and the response of plant to the relative length of light and dark period is called photoperiodism. Garner and Allard in 1920 first established the phenomenon of photoperiodism in Maryland mammoth variety of tobacco. On the basis of photoperiodism requirement, flowering plants are classified into short day plant, long day plant and day neutral plant. Short day plant are these plants flower or produce flower when they receive photoperiod shorter than a critical period. They are also called long night plant because they need an uninterrupted long night for flowering. The most of the winter flowering plants belonging to this category. For example, soybean, sugarcane, dahlia, chrysanthemum, rice, tobacco and xanthium. Then long day plant which require a long day or long light period and short night period for flowering. They can flower even under short day if long dark period is uninterrupted by brief flashes of light. They can also flower even under short day if long dark period is interrupted by brief flashes of light because they require a short night period. These plant flower during summer such as in spinach, sugar beet, radish, plantago, wheat, barley, clover and lettuce. These are long day plant. Then day neutral plant. These plants are photoneutral, that is they do not need a particular critical period for flowering. They can flower throughout the year. For example, cucumber, cotton, tomato, potato, sunflower, garden pea, maize, chili, etc. So it is all about the photoperiodism. Then what is phototropism? Phototropism is a phenomenon by which the plant bends in the direction of light. So suppose it is the light source and it is the stem or shoot apex of the plant. So accordingly or gradually it will bend towards the source of the light. And this process is known as phototropism or tropic movement of the plant towards the light source. Charles Darwin experiment. At first he did an experiment by using canary grass and oat coleoptile and recorded his observation in a book, The Power of Movement of Plant. They observed the bending of seedling towards sunlight. They demonstrated by covering the tip of the oat coleoptile, which prevent it from photosynthesis. Simultaneously, when they covered the lower portion of this coleoptile, they turned phototropic. That means only the apical part grows towards the light source. He concluded this from this and several other experiments that the tip of the grass, which is known as coleoptile, have strong sense of light and thus bend towards the source, while the middle section activates proton, decreasing the pH of the cell. This act acidifies the cell wall, activating the enzyme known as expansion, which break the cell wall, thus making less rigid or flexible. So they bend towards the light source. Then Boyson-Jensen experiment. Reflecting on Darwin's experiment with oat coleoptile, Boyson-Jensen in his experiment in 1913 experimented on the seedling by cutting off the tip and replacing it with thin layer of gelatin between the tip and the cord stem. This did not prevent the stem to curvature towards the source of light. But when he placed a small mica sheet, he concluded that it prevents curvature. Then the scientist placed the same mica sheet on illuminated side. There was no curvature. He concluded that a flow of material, later named as oxygen, which passes through gelatin and mica sheet, was caused behind the curvature. That means at first he caught the coleoptile and placed a gelatin. Through gelatin, that chemical can pass, and he observed the bending towards the light source when he placed the gelatin. Later on he placed a mica through which the oxygen or any chemical cannot pass. Later on he observed that there is no curvature of the coleoptile towards the light source. Then he placed this mica sheet. So when Boyson-Jensen placed the mica sheet in the illuminated site, so after placing the mica sheet in the illuminated site of the stem, it does not prevent the curvature. That means curvature occur. But when he placed the mica sheet in the darker side, it prevents the curvature of the coleoptile. That means he concluded the oxygen or the chemical that causes bending actually formed in the darker side of the coleoptile. He concluded that a flow of material, later named oxygen, which passes through gelatin and can't pass through mica, was the cause behind the curvature, and it was later on discovered as oxygen. Then the phototropism mechanism. What is the mechanism behind this phototropism? The mechanism of phototropism is as follows. Light of wavelength for 450 nanometer or other illuminate the plant, the photoreceptor receive the light, react to it and initiate the response. Phototropins are the proteins that receive the blue light during phototropism, generally receive the blue light. Auxin moves a darker side in the stem when exposed to light, and it is generally produced more in the darker side, so causes bending towards the light illuminated side. Auxin releases hydrogen or H plus ion in the shaded region of the stem, which causes the decrease in pH. This decrease in pH activates the expansin that causes the cells to swell and forces them to bend towards the light. So this is how phototropism or tropic movement or movement of the shoot tip towards the light occur. So it is all about the plant growth hormones, photoperiodism and phototropism. I hope you really enjoyed this session. So stay tuned to get more videos like this. Thank you.